Hello and welcome to STR IAS. In this video lecture, we will be looking at a topic called human reproduction for UPSC. So purely from the UPSC point of view, we will be discussing about human reproduction, and we'll be looking at what UPSC has asked previous years, and most probably what happens if mitochondria of mother is having a defect and all. We'll be looking at, because the same theme has been repeated for almost 3 to 4 times, so it will be a matter of importance, right? So without making any late, so we'll get into the topic. So humans, we know that humans are made up of cells. So in that cells we'll be having DNA. So DNA will be condensed into chromatin, then chromatin will be condensed into chromosomes. So chromosomes will look like this. So in this shape is called telomere shape. Okay, so what will be there in DNA? So DNA will be having characteristics of species. So DNA will be having some information which defines the characteristics of species. For example, human should have 2 hands, 2 legs, 1 head, 1 heart, kidney, and all. These characteristics are defined in are mentioned in, or information is available with DNA in the form of chromosomes. So how we'll be comparing, or how the characteristics will be, we can see through this image. For example, compare the DNA of human and compare the DNA of rice. It will be different, just because rice will be having 24 chromosomes and human will be having 46 chromosomes. When it comes to roundworm, 4 chromosomes it is there, and when it comes to chimpanzee, you can see that there are 48 chromosomes, almost nearer, right? So chimpanzee will be almost nearer, or it will be looking almost nearer to a human, right? So DNA will be having characteristics of a species. DNA will be having chromosomes which define the characteristics of species. So these chromosomes, which is a condensed or coiled form of DNA, right? This will exist only as a pair. It will be existing only as a pair. For example, we have 46 chromosomes in a human being. So it is nothing but 23 pairs of chromosomes, and each pair is denoted by n, right? So we can see like 2n. It means 2 into 23 equal to 46, right? So if it is like only this is called 2n is called, configuration is called diploid. So human will be having 23 pairs of chromosomes. If a cell has only one set of chromosome, then it is called haploid. If it is only one n, it is called haploid. Then why chromosome will exist in pair, or why there will be haploid cells, we'll be looking at it. So cell, so for anything to grow, they have to divide. For example, one cell, if we have to grow, it will be dividing like this, right? Then from here it will be making a daughter cells. Then again daughter cells will be same. It will be happening like this. Then some more daughter cells. So the cell will be dividing for 2 functions. One is for growth, another one is for reproduction, right? Generally it happens. Then why pairs? So for example, mother will be having 46 chromosomes. Everyone's mother will be having 46 chromosomes, and every father will be having 46 chromosomes, and the baby will be getting 23 chromosomes, not 23 pairs. You should just look into it. 23 chromosomes from mother and 23 chromosomes from father, so making it 46 chromosomes totally. 46 chromosomes, not 46 pairs, 46 chromosomes totally. It means that the baby should get half from mother. Half means 23, right? And from father, 23 chromosomes are needed, not 23 pairs. 23 chromosomes from mother and 23 chromosomes from father should be needed, so that baby will be getting 46 chromosomes totally, and it will become 23 pairs, which gives human characteristics, right? So I need only 23 from my mother and 23 from my father, not pairs, chromosomes, but both have 46 chromosomes. So from that, so we should get only half from the each 23 chromosomes and 23 chromosomes, right? So there will be some cells in the human body which will be having 23 chromosomes. I mean, half DNA only will be there in that cells. Those cells are called gametes. Those cells are called gametes. In the male, the gametes are called sperm cell, and in the female, the gamete is called ovum or egg, right? Then, so half only will be there. So sperm cell will be having 23 chromosomes, ovum will be having 23 chromosomes. Totally, if it combined, will be getting 46 chromosomes, and a new cell is formed, a first baby, right? So it is called zygote. It's called zygote. So observe, we have two types of cell divisions. I told cell division primarily happens for two reasons. One is for growth, another one is for reproduction. So if it is for growth, it is called mitosis. It is called mitosis. For reproduction, it is called meiosis. Okay. So for growth, it is called mitosis, and for reproduction, it is called meiosis. So mitosis will be producing diploid cells, 2 and 2, right? And meiosis will be producing haploid cells. Haploid means N, right? Mn. So mitosis, for growth we need 2N, and for meiosis we need only one single N. Then we need all the DNA here. From cell have to divide, and for the growth we need all the DNA, but here for the reproduction we need only half DNA from each cell from mother, half DNA from father, half DNA will be getting, right? So we can see, so 2N just becomes 4N, then it will be converted into 2N and 2N, right? So we can see here 2N will be converted into 1, 2, 3, 4N, then at last it will be converting into 2N and 2N. So in meiosis, just see here. So parent will be giving us half DNA. I mean, parents will be having 1, 2, 3, 4. So parent will be having 46 chromosomes. I mean 46 from each mother and father. Nothing but 23 pairs from mother and 23 pairs from father. Then it will become 2N first. Then it will become 2N plus 2N. Then after that we can see that, so here it will be divided into single N, single N, single N, single N. So it will be making 4 daughter cells. Here it will be making only 2 daughter cells, right? So if you are not understanding, just remember mitosis, 2N in everything. So diploid cells. In daughter cells also will be having diploid cells. These for growth, right? So when it comes to meiosis, this type is for reproduction, and in this haploid will happen, and we can see that 4N cells, I mean N plus N plus N plus N, it means haploid will be happening here, right? So this for growth. Meiosis N, mitosis 2N. In mitosis 2, 2. Okay, simply you can remember that, not to get confused. Then we have seen gametes. So in the gametes, the male gamete is called sperm cell. So the sperm cell structure will be like this, alright? So sperm cell will be having head, middle piece and tail. In the middle piece will be having mitochondria, which will be powering for this action. It will be moving, so it should penetrate into egg, right? It should be moving, or it should be getting inside to the egg or ovum. So it should be it should be moving. For that the energy will be given by, and DNA will be present here, right? And this head part is called acrosome, so no need to remember all those things, right? And the gamete, the female gamete is called ovum or egg. You can see that there is nucleus and everything which is there in the cell, it will be there in gamete. Then fertilization. So there is one sentence or one dialogue in Three Idiots. So there will be 3 million or some millions of sperms. In that, only one sperm can make it. We are already a winner, right? So many of the sperms will be reaching the egg with their, when I was teaching about cell membrane, I told it has phospholipid bilayer and it won't allow all the things inside. So there will be two types of transports, active transport and passive transport. So to get into this egg, the sperm needs something called active transport, nothing but energy. Energy is needed. So that energy, with that energy, it will be entering into the egg and fertilization happens. So this is the way. So the sperm will be entering through vagina and it will be entering like this, and egg will be coming here, and this tube is called fallopian tubes, right? So here the fertilization happens. Fine. Then zygote, the first cell after fertilization happens. The first cell which takes birth after the fertilization happens. So zygote is half from mother and half from father. So mother will be giving all the cell organelles like mitochondria, then Golgi apparatus, then vacuoles, will be having endoplasmic reticulum, cell membrane, nucleus, everything will be given by mother. Then half from the father. So in the zygote, half from the father, and it will be only nucleus. So it will be only nucleus from the father, and we'll be having everything from our mother. We'll be getting everything from our mother. So from mother we'll be getting all cell organelles. From father we'll be getting only nucleus, right? So think that. So here it is father's nucleus, it's mother's nucleus, mother's endoplasmic reticulum, mother's vacuole, mother's mitochondria, mother's Golgi bodies. So what if the mitochondria of mother has some defect or problem? So mitochondria is also called as powerhouse, right? So powerhouse for what? Powerhouse for everything. It will be giving energy, or it will be providing for every energy for every function. What if mitochondria of mother has some defect? So there is a question here, which is another organelle other than nucleus which has DNA. So other than nucleus in which has DNA, which is the another organelle in the cell. So it is none other than mitochondria, right? So in prokaryotic cells we will be having plasmid, which will be having extra chromosomal DNA, but in eukaryotic cells mitochondria will be having DNA other than nucleus. Then what if mother's mitochondria has a problem or defect? Mother can survive, just because mother has survived already 20 years or 30 years, right? So she can survive, but what about the baby? What of the baby like which will be getting birth? So the thing is, baby will be getting a disease called Leigh disease. The baby will be getting a disease called Leigh disease, and this type of disease are more common due to mutation, or mother's mitochondria will be having a problem, right? This is mostly due to consanguineal marriages. Nothing but if you are marrying like cousin marriages are more prevalent in South India. For example, a girl marrying mother's brother, right? So same DNA, the variation of DNA should be there. DNA variation should be there. If there is no variation in DNA, then this type of problems might occur. So in Leigh disease we can see the symptoms. So psychomotor aggression, no brain development will be there, and encephalopathy, it means encephalopathy means brain development will stop. Then hypertonia, then deafness will happen. We can see that pigmentation on eyes, right? And failure to thrive, or like always will be prone to infection, right? So this happens for new baby. So new baby will be having a problem since inception, nothing but since the birth, no proper development will happen. So is there any possible solution to avoid this? So possible solution is nothing but prevention is better than cure. So one of the solution is mitochondrial replacement therapy. The name itself says that replace the problematic or defective mitochondria which is there in mother. And where we'll be doing it? We'll be doing it in lab. It is called in vitro, right? So baby's mitochondria completely comes from mother, right? That's why we need to change the mitochondria of the which is present in mother or in zygote. So from where we'll be taking a mitochondria? We'll be taking mitochondria from the third person called donor. If a donor has a healthy mitochondria, we can get from them. So first method of mitochondrial replacement therapy is pronuclear transfer, right? So we will be having biological parents. So father and mother. From father will be getting sperm, and from mother will be getting ovum, and this from father will be getting sperm, and from mother will be getting ovum, and this mother will be having mitochondria problem, right? So here we can see that father's nucleus, and this is mother's nucleus, and we can see this everything is mother's, and this is problematic mitochondria which is of mother's, which is formed as a zygote. Then we'll be getting healthy mitochondria from donor female, right? So we have a donor who has ovum in which we have a healthy mitochondria. We'll be removing this, we won't be removing that, we'll be removing this nucleus which has formed, right? So it is a pronucleus. Pro means old or at the beginning stage, right? So remove this pronucleus and add into the cell of or ovum of donor. Ovum of donor. So it will be having healthy mitochondria, healthy endoplasmic reticulum, and we'll be adding this. This is of fathers, and this is of mothers, and this cell will be of third person donor. That's why this baby is called 3-person baby, 3-parent baby, right? So we are transferring the pronucleus. That's why the technology is called pronucleus transfer. So we are removing the nucleus of donor and we are adding the pronucleus of mother and father. So pronucleus transfer. Then plant this egg which is of donor's egg with healthy mitochondria, healthy endoplasmic reticulum, healthy vacuole, but the nucleus will be of biological mother and father, and plant this egg into the biological mother, and she'll be getting a baby who will be having a good or healthy mitochondria. So this pronuclear transfer happens after fertilization. So once the sexual activity happens, so sperm and ovum get fertilized and zygote is formed. In zygote, in the primary stages, we'll be having pronucleus, like nucleus from sperm and nucleus from ovum. So this pronucleus will be adding into donors. So this pronuclear happens when after fertilization, right? So I have told that already. This is called 3-parents baby, because we'll be getting mitochondria from donor. We also seen that mitochondria is the only organ or one more organ which will be having DNA. DNA is nothing but characteristics, right? Characteristics in the form of chromosomes. Then what about can we do it before fertilization? Of course yes. So we can do this technology, we can use this technology before fertilization also. So before fertilization, so there is a biological mother and we have donor healthy female, right? So just take this nucleus and remove this nucleus, remove the nucleus of donor, who have healthy mitochondria, and add this nucleus of the mother into the ovum or egg of donor. Then take the sperm of father and biological father and fertilize it in the lab. So this happens before fertilization itself. So before fertilization itself, we are changing the mitochondria. We are changing everything. Literally, we are taking only nucleus from mother and we are fertilizing with the help of fertilization happen after we are changing the nucleus, right? So this is called reconstructed egg technology, and it is also called as maternal spindle transfer. So once again we'll revise. So we have two methods. First one is pronucleus or pronuclear transfer. Pronucleus transfer. It happens before fertilization. It happens after fertilization. After fertilization, nothing but after the sexual activity, the sperm plus ovum gets fertilized and will form zygote. In zygote, we'll be having pronucleus, nucleus of mother and nucleus of father, right? And we'll be having defective mitochondria. This defective mitochondria should be replaced. That's why what we'll be doing, we'll be taking this pronucleus and we'll be adding into the donors, right? So this will be replaced here, right? So this is called pronucleus transfer. This also can be done. Before fertilization. And this method is called maternal spindle transfer, or it is also called as reconstructed egg technology. So these are the things which are more frequently asked in UPSC, mostly about mitochondrial diseases, what is Leigh disease, then what about in vitro fertilization, what about like maternal spindle transfer, why we call it as 3-parent baby, and we can look at like from the mains perspective also, like issues like surrogacy and all will be asking from this topic. So hope you have understood.